नमस्ते। आई एम डॉक्टर खगेंद्र कुमार, DRG गवर्नमेंट गर्ल्स कॉलेज मल्होत्रा। सेकेंड ईयर के लिए मेरा एक टॉपिक फर्टिलाइजेशन। फर्टिलाइजेशन इंट्रोडक्शन। Fusion of two types of sex cells or gametes is defined as fertilization, in which union of the nuclei is referred to as karyogamy and fusion of the cytoplasm of the uniting gametes is defined as a cytogamy. Complete fusion of two gametes is described as a syngamy. Types of fertilization. External fertilization. If fertilization occurs outside the body of the female parent, then it's called external fertilization, in which water and is required essentially because female releases its ova into the water, and that releasing process is called spawning. Similarly, the male also releases its spermatozoa in water. For external fertilization, water is very essential. और एक्सटर्नल फर्टिलाइजेशन के अंदर जो फीमेल है वो एक समूह के अंदर बहुत संख्या के अंदर अपने एग रिलीज करती है, और मेल भी स्पर्म एक समूह के अंदर उन एग के ऊपर रिलीज करता है। यहाँ पर फर्टिलाइजेशन की चांसेस कम रहते हैं, इसलिए नंबर ऑफ ओवम और स्पर्म ज़्यादा होने जरूरी है। एक्सटर्नल फर्टिलाइजेशन मोस्टली इनवर्टिब्रेट्स एंड एक्वेटिक वर्टिब्रेट्स के अंदर होता है, जैसे टीलियोस्ट फिशेस एंड एम्फीबियंस। इंटरनल फर्टिलाइजेशन इन मैनी एनिमल्स टू डेफिनेटली ensure fertilization. The spermatozoa are internally deposited by the male with the help of copulatory organs into the oviduct of the female, where fertilization takes place. The process of deposition of spermatozoa in the oviduct of the female is called insemination. That's why it's a surety of a fertilization, and chances of a fertilization of eggs are higher in the internal fertilization. On the basis of the site of embryonic development, animals are categorized into three groups: oviparous, oviviviparous, and viviparous. Mechanism of fertilization. First process, approach and penetration of spermatozoa to ovum. Second, activation of egg, and third, fate of spermatozoa inside the egg. In mammals, it will see that in first process, sperm inactive stage के अंदर होता है and ये favorable condition के अंदर activate होता है. That process is called capacitation. Protein present on a sperm head is called anti-fertilization, which is glycoprotein, and protein which is present on ovum surface, that is fertilization. There is a reaction between sperm and ovum called fertilization anti-fertilizing reaction. Here you will see that part of a fertilizing particle of a fertilizing which is present on ovum, and also binding of fertilizing and anti-fertilizing. In process, first process, contact. The sperm cell contact the egg's jelly coat, triggering exocytosis from the sperm's acrosome. Acrosomal reaction started. Hydrolytic enzymes released from the acrosome makes a hole in the jelly coat, while growing actin filaments from the acrosomal process. This structure protrudes from the sperm head and penetrates the jelly coat, binding to receptors in the egg cell's membrane that extend through the vitelline. Third process, is contact and fusion of a sperm and egg membrane. A hole is made in the vitelline layer, allowing contact and fusion of the gametes. Plasma membrane. The membrane becomes depolarized, resulting in the fast block to polysperm. Entry of a sperm nucleus, and the fifth process is formation of a cortical granule. In process of a cortical reaction, fusion of the gamete membranes triggers an increase of a calcium ion in the egg cytosol, causing cortical granules in the egg to fuse with plasma membrane and discharge the air contents. This leads to swelling of the pre-vitelline space, hardening of the vitelline layer, and clipping of sperm binding receptor. The resulting fertilization envelope is the slow block to polysperm. As well as we know very well that a sperm head contains some hydrolytic enzymes which are very essential to enter in the egg membrane, and that hydrolytic enzyme is one example is hyaluronidase enzyme. In next slide, if you will see that sperm head contact with the egg membrane. Second, acrosomal reaction, when the sperm head releases hydrolytic enzyme known as hyaluronidase enzyme, and then the actin filament formation, acrosomal process, and fifth process is entry of a sperm nucleus, sperm head, and proximal centriole, and fertilization membrane form. Here you will see that binding of spermatozoa to zona pellucida, and acrosomal reaction. Third, penetration through zona pellucida, fusion of a plasma membrane, and sperm nucleus enters. Block polyspermy. After entry of one sperm in the ovum, now it's very necessary and important to block polyspermy. To block polyspermy, early step is sodium ion present on plasma membrane go inside and depolarize the membrane. Now conformation changes, and that's why sperm will not enter. And late step in which binding receptor activates P1 P2 phospholipase C, then release IP3 inositol 3 phosphate and DAG diacyl glycerol. It activates phosphokinase C, activates sodium proton pump present on plasma membrane, and pre-deposited mRNA and protein get activated. pH increase on plasma membrane and sodium ion. That's the result of membrane depolarization and stop entry of sperm. Another process on sarcoplasmic reticulum, calcium ion concentration increase, cortical granules fuse with plasma membrane containing water absorbing substances, protein, fertilization, envelope formation, thick coat, and removal of all remaining receptors with the help of protease enzyme. And in genome imprinting at the genomic level, it will decide whether the male gene will express or female gene will express. Types of epigenetics. Methylation will be seen to deactivate the one gene expression. Rotation of a cytoplasm present, receptor present over the animal pole in frog. Here you will see that Greek recent found, and it will play a very important role in differentiation, determination, and morphogenesis. And rotation of Excitoplasm is called cortical rotation. Gray gradient is very important for further development. Here you will see that GSK3 काइनेज जिस तरफ प्रेजेंट होता है वो वेंट्रल साइड और बेली का फॉर्मेशन करवाता है, और बीटा कैटेनिन जिस तरफ ज़्यादा मात्रा में प्रेजेंट होता है वो डोर्सल साइड और बैक का फॉर्मेशन करवाता है, और बीटा कैटेनिन हमेशा उस तरफ प्रेजेंट होता है जिस तरफ ग्रे क्रिसेंट है वो प्रेजेंट रहता है। ग्रे क्रिसेंट के अंदर ग्रेन्यूल्स कंटेन प्रोटीज एंजाइम टू which inhibit the GSK3 kinase fused after rotation, and protein continuously produced in egg by that is beta catenin phosphorylated by GSK3 kinase and degradation of beta catenin. बीटा कैटेनिन का डिग्रेडेशन होता है तो फिर वहाँ पर GSK3 काइनेज ज़्यादा मात्रा में होता है और वेंट्रल साइड का फॉर्मेशन उसके अंदर होता है। थैंक यू। कुछ स्टेप बाकी रहे हैं उसको अगले वीडियो के अंदर अच्छे से समझाया जाएगा। थैंक यू सो मच।